Are live. Fantastic. Okay. Welcome to the first CNS talk of 2025. I'm super excited to have Tim Dyrby with us today. We had to push this talk back twice, I think, but most recently by a week for very exciting reasons. And we're going to get a little preview of what Tim's been up to last week. Little bit of background. Tim has a solid background in engineering and informatics as his undergraduate, postgraduate, and PhD. And he is currently a professor and group leader for microstructure and plasticity at the Danish Research Center for Magnetic Resonance at Copenhagen University Hospital, and also part of the visual computing section at the Department of Applied Mathematics and Computer Science at the Technical University of Denmark. Tim, without stealing any of your time, take it away.
Thank you, Stephanie. And thank you for this invitation to make this talk here. So it's good to see all of you guys. And I put some slides together for what we've been working on in my group for the last many years. And it will be a mixture, a lot of technologies. And you can see from the title is how do we actually measure stuff? And my focus will be about conduction velocity along axons. So let's get started. If I can change slides here. So we will start from the basics. So we all know we have this brain here from the MRI with the magnetic resonance imaging here. This is living brain, it's in millimeters. And this is just a T2 weighted image where we can see everything 3D of the gray matter and white matter. But what we know is that in white matter, we have these pathways here. And it's just illustrated here for some of the one going from the motor cortex down here to change where we actually can make our movements, legs and arms for the cortical spinal tract. And then we have these fibers here. This is example of a callosal fibers, big bundles connecting the two hemispheres. These we cannot see on these MR scans here, but with other techniques, we actually can zoom in. And what we know actually here is that between these connections here, we have the input and we have the output of the neuron here. And what is the white matter is these myelinated axons here. So we can zoom in on these axons here in a bundle within a small voxel. And then we will see this massive population of axons that are making this conduction velocity along the brain. And there are different techniques to see it. This is two different ways of seeing with electron microscopy. And if we take the cross-sectional here of these axons, then we can see here that they have these black rings around, which is the myelin. So the cell membrane of the axon is within this area here. So it's a very dense area in white matter of these myelinated axons. That in the primary brain, there's a fewer of these unmyelinated axons. What has been an interest for many years is to look at this distribution of the axon diameters. So how big are they in diameter? And this is one study here from Caminiti and Innocenti, where they have been looking into the human brain in corpus callosum to see how big are these axons here. And this is just a distribution. We can see typically the peak is around 1 micrometer, and then we have a distribution of diameters going up to 5-6 micrometers here with like a tail. So this is what we call it the mode, that is this peak here. And if you compare with different subjects, you see the different colors, then they have some kind of same range if you measure in the same organization. So why is it that we are interested in these axon diameters? That is simply because that we have in these pathways in the brain, we have these connections. Some of the short ones and then the long ones. And they are controlling everything what we're doing, our thoughts, our motor input and output. And one thing is especially important is we get this idea that we want to run for simple reason. That could be we are threatened somehow. It's pretty important and we can react and get our legs to move on when we get this first thought. So therefore we have this long connection from the motor cortex sending out the orders down here to the legs, and these need to be fast. So this, how fast the signal are what you can see conducting along the axons, this is determined by the structure of this axon. So we have the signal coming down here from the soma, firing down to the dendrites. So many of these structural features here that influence on the speed, how fast the signal can travel along the axons here, that has been looking in for simulation studies. And there's a very comprehensive study here by Dragsmith from Cardiff. And he has been looking on myelinated axons. And one of these factors that actually really matters on the conduction velocity is the axon diameter. Others are the node density, because the axons have like nodes, so sheets, and there's a small distance between these sheets of axon between the myelin, and that also matters. And those lengths and all these kind of features also matter of the conduction velocity. But one of the driving factors are specifically the axon diameters. So if we now look into this more at detail, so we can make this simple to what is the relationship then? So if we just taking from the previous study here, what are the features that really matters for conduction velocity, then we can map here that the axon diameters here, they get smaller and larger here. And they actually have a kind of linear relationship to the conduction velocity. So the bigger axons, the faster the signal actually will conduct along the axons. And then we have, of course, also have the myelin thickness here, which changes with the size of the axons. So this is an example of a normal axon here, where the signals are transmitted along the axons. We can see the signal here going down here. But what if we now have the situation of pathology? So we can imagine we have a demyelination disease, like it could be MS or something, making a focal demyelination along the axon. What is then happening to the conduction velocity? Well, that's a really good question, but we know it will change. And why it will change is simply because that myelin is one of the driving forces in the speed of conduction velocity along the axons. But if we really want to study it, there's a few studies showing that if you lose the myelin around the axons, then there's likely that you will have a blocking of the conduction velocity for a certain period. Nevertheless, we can actually, from these structural features, start to see, can we predict conduction velocity? We can do it in two ways. We can do it from the structure. So this is the conduction velocity. Then we have this formulation that is established by empirical studies, where we have a factor 5.5 G. This is the G ratio. So the G ratio describes the ratio between the outer diameter here plus the myelin and the inner diameter of the axon here. And if you take this ratio between here, that is then the G ratio between the myelin and the axon. Then you have D, that is the diameter of the axon. So if you take this 5.5 divided by G and D, then you get the conduction velocity from the structure point of view. And the G ratio here from many studies is around 0.7. And that is something you can do a lot of simulation, and you will find out that this is the optimal range of conduction velocity along the axons with this G ratio. Now we can also actually predict the conduction velocity from latencies. So we could have a stimulation. We can stimulate the neuron here, and then we will initiate a signal transmitting along the axon, and then we can measure the time it arrives at the target area here, down in the dendrites. So if we have this latency measured here, and we know the length of the distance here, then we can also calculate the conduction velocity. So the big question is, how does these parameters actually relate to each other when we predict either conduction velocity from the structure or conduction velocity from the latencies? So we want to be able to do this, we need to measure the G ratio. We just need to have the axon diameters and the myelin thickness. And for the latencies, we just need to actually measure the conduction, the latencies here, and know the length of the distance here. And there's different ways of doing that. This is an example from Giorgio Innocenti, a very nice study here. So what they did was to predict the conduction velocity in the brain from the structural features. So they did injections in the macaque monkey with a BDA labeled tracer. And this BDA labeled tracer, you can actually see the labeling on electron microscopy. So this is, it's appeared dark. This is an example of labeled axons with this BDA. So you can actually see those axons that come from the injection site to their protection site. What they did was cutting this brain into many fine slices. And then Giorgio, he was sitting in his microscope and actually drawing each of these small labeled axons projection from the injection site to wherever it target in the brain. It's a massive work, and you can see this is an example where you can see how the small segments of axons that are labeled in each of these slices appear. He has been drawing to see where the target. And from this here, he also can detect the axon diameters. So this is an example of a small zoomed in where you can see this is the axon diameters. So these are the ones that are detected in these when you trace through these trajectories in the brain. And what they show here is that along one fiber track, you actually can see that when you go from one area, this is area 4, the primary motor cortex, then you can see the axon diameter distribution actually changes when you go along the track. And that is probably because you have different tracks. It's a main pathways, and then you have some tracks that is bending off at the different projections. And that will change this size distribution of axons in one region of the along the track compared to others. When you have these axon diameters at the different positions in the brain along the pathways, then you can make this prediction map of the conduction velocity. And all these are injection sites, different regions in the brain. And we can see here, this is corpus callosum. So if you measure the axon diameter in corpus callosum, for example, from the motor areas here, area 2 or 4, or you can say the prefrontal regions here, you can see they all project through corpus callosum, and that we know. What this drawing shows is that the broader the peaks are, the thicker axons. And the number here describes the conduction velocity. So when we have a very thin axon, it's the thin line here is small axons, and of course the conduction velocity then also is slower. And this is what we expect because this is the motor pathways, which are very fast. So they are thick and very fast, whereas the prefrontal, which we know is slower, they have smaller axons with a slower latency. And it's the same thing if you follow this area 9 in the prefrontal down to the internal capsule or thalamus areas, you can see it's all small axons. Whereas if you have the area 4 with the premotor cortex going to internal capsule, where we have the cortical spinal tract, then it's big axons following down there. So that makes sense. But when we want to image these kind of microstructural features, then we can do it in different ways. So this is an example of image resolution here. And then we have anatomical features out here. So what of this line here is technologies. This is diffusion MRI, and this is different microscopes. So microscopes can go to much higher resolution. So they can map cells and other features here in the micrometer regime, so we can see them directly. Whereas if we go into the MRI, we cannot, because of the low resolution, really see them directly. So what we are seeing here is that we use a model to predict the sizes of these anatomical structures we want to see. So what we can see is here that if we are in this millimeter regime here, then we can actually map out the organization of the brain. But when we go into the finer regime here, in these in micrometer and submicrometer levels, then it's the structure of the features of the cell sizes or the axons and myelin that we're actually interested in. And here we can measure these directly with light microscopy, electron microscopy, micro-CT, and we can actually also see it with diffusion MRI. So do all these methods here show the same thing? So that is the question. So if we now start to look here for different 3D imaging techniques that we will be looking at. And why I say 3D imaging techniques now is because I want to move a bit away from the 2D electron microscopy and what is traditional, because what we are looking at here is 3D structures, but I will show you later. So we have as a typical setup here. So we have a brain, and if we want to measure this brain here, the microstructure, then we can use an MR scanner, 3D MR scanner. It's not because it's 3D, but it's measuring the structures in 3D. And here we have this diffusion MRI scans we have, and we can fit a model to these data sets, and then we can predict the size of the axons. We can also image these features here directly using microscopies, and this is often what we have this knowledge about how should our model look like. These models here, they're coming from these assumptions here. And what I'm going to talk about today is this 3D synchrotron, X-ray synchrotron imaging techniques, and there are also possibilities for 3D EM. So when we start to use these techniques here, we need to process the tissue in different ways. And that matters when we are going to compare techniques of the results. So if you look at the pipeline, so we have the MR scanner here. This is the in vivo. We can put patients in it. We can have animals or whatever. We can scan them in vivo. But of course, there are some limitations in the resolution. Then we can fixate it. Not always the patients, but we can take the animals and fixate these and scan the brains here. And there we can do it post-mortem with the MR scanner. We can then process the tissue more. Typically, you embed it in the agar, you start to cut it down, you can puncture small areas, and then you can osmum embed it to actually see the myelin or do another kind of staining to see it in high resolution. Then you often can embed it in paraffin, or you can embed it in EPON, and I will only talk about EPON embedding here, plastic embedding. When you have plastic embedded it and stained it for myelin, this is osmostain, then you can do your electron microscopy here, or you can start polishing your sample and make it round, and then you can go to these big X-ray synchrotron facilities to have it in 3D imaging. But when we process this tissue here, then we need to aware what is happening to the tissue. So when we come it out here from we have the fixation, then we have the tissue here for MRI, and that is hydrated tissue. So that is a state which is very close to in vivo state. It's of course fixed, but it's still hydrated with a lot of water. When you start to process your tissue to EM or to synchrotron imaging, you often go through a stage which here is dehydrating tissue step. And this is what you do with standard EM. And if you do that, then you in this part here risk to have shrinkage effect because you dehydrate the tissue. Alternatively, what you can do is to do high pressure freezing, cryo-EM, but that only works on very thin slices here. And then you will not introduce this kind of shrinkage. And in that case, you will have something that more match what you would expect to see on MRI. So this is something we need to be aware of in the tissue processing. So what about the tissue quality? So that is also important when we go to postmortem. It has been shown that in the tissue, this is an example of electron microscopy from tissue from a tumor operation. And after a few hours, you actually start to see the myelin layers loosening. So this is after 6 hours. So the myelin loosening and the disruption of the tissue is going very fast. And if you want to slow it down, then you need to make you need to keep your tissue cold. But fixation needs to be very initiated early after death. Otherwise, you will get this autolysis effect. This is one of our own example with Maurice, where we have a monkey brain where something failed in our perfusion. And here you can see a totally, completely disrupted axonal environment. You can see the cell membranes here, and all the myelin layers are completely loosened around it. So this is not an anatomical environment we can compare with in vivo. This is when you do it perfusion fixation, where everything is working. Myelin is nicely wrapped around the axons, and it's very coherent environment. So this structural environment should look very much what we expect to see in vivo. So one thing about this with the human is that you always somehow, when you do larger sample, will have this post-mortem interface. So this is examples what we have been doing on synchrotron. So this is a larger sample taken from biopsy of a human brain that have been stored for many years. They, in the report, wrote that they took the post-mortem interval of 60 hours. So this is not an unusual brain you investigate for human experiments. But when you look at it in 3D, so this is 3D volume, you can see large spaces here. And if you try to follow the axons, you cannot see it here. You will completely look in 3D that these axons look like some kind of balloons that are sometimes exploded piecewise. And that is not normal. And this is when we zoom in, you can see the myelin is disrupted in layers. And yeah, it's not normal. But if you fixate it with short postmortem interval, so this is a human we just collected here. This is 5 hours postmortem interval. Then you can actually see it's a bit zoomed out, but you can see all the axons here and the dark stripes. That is the myelin. This is nicely tightly around all cells here. You can see the cells and the cell membrane of the glia cells, where there are small intercellular structures in here. The very fine details are preserved. So the quality of tissue and postmortem interval is very important to keep normal. Then we have also this step that I was mentioning about the tissue processing. So we have the standard EM here, where we have the dehydration step, and compared with the cryo-EM. what you see here is not because we have a contrast difference, but what we see here is that in the standard EM, you can clearly see that all the axons here, they're very closely, the density is high compared to a cryo-EM here. So here we take a small piece of tissue and we high pressure freeze within few milliseconds. So we preserve the whole cellular environment. So what you see here is that in this case here, that a lot of the extracellular space is preserved, which is like disappear here in the dehydrated situation. If you now compare these two setups, so this is in the mouse, like the rat brain it is in corpus callosum here. So this is a light microscopy. In this region here, we took out samples for standard EM, and this is for cryo EM. So what you see here, this is automatic segmented, and these are manually segmented because it's a more difficult, but we have a method now that can do this automatically also. So if we can compare here these two environments, we can see that the axons here, they are the density is high compared to down here with the cryo-EM. It's the same regions. But when we also look at the histograms of the two here, then you can see the peak here, the mode of the distribution here in the standard EM is a bit lower compared to the one in the cryo-EM. So we have a shrinkage here. When we look at the scaling here, the X scale is not so optimal. So you can see this is 1 micrometer. And if you compare it here, this is 1 micrometer in the cryo-MA. So you can actually see we have some kind of severe shrinkage of the larger axons compared to the smaller ones. And if you look closely in here, then you can actually see we have like a second mode, or we call it a shoulder. It appears sometime on the distributions. What you see here is that that here this is around in this area, and you have this is massive over 1 micrometer in this. So we have this shoulder here is about 48% of shrinkage, and the small axons here, they're around 33. So this gives an average about 40%, which is typically reported, but it's very broadly set what people think of what shrinkage is. It's from 0 up to about this 40-50%. But it looks like axons shrink in different ways, smaller, less than larger ones. We can then also estimate the G ratio here. This we, for now, I only show for the standard EM, and this is a number of animals, and you can see they all overlap here with a G ratio of about 0.7, which is expected from the literature. But all literature is also based on the same way of electron microscopy. So G ratio, and we have some shrinkage we need to correct from. So next part is, when we want to analyze these axons here, then we make this drawing, sketch. There's a nice straight axon, and this is a round circle where we have axon in here, we have myelin layers. We can do in corpus callosum, and we can do our things here with electron microscopy, and you can see we have a change in the axon diameters, what we also expect to see here across corpus callosum. And axons seems to have a different shapes. So what we then want to quantify here, so what I would say here is that we also see axons here that actually not only go in one direction in corpus callosum, but you also have axon crossing here. This is one you can see an example here in the 2D here. So how do we quantify this? So in this case, and that is what we always have been assumed is axons, they are cylinders, which is not true. But that's for now what we believe. So we can measure here it in two ways. We can take the diameter, calculate by elliptic minor axis. So we make an ellipse here, and then we take this axis here to be, that is a true axon diameter. Or we can take the equivalent diameter where we have the area here, and then we calculate the corresponding diameter. And when comparing these matrix here, this one here is more conservative than this matrix here. But why do we see this kind of change in the shape? And that's of course, if an axon is not completely aligned with what the field, then it will have a shape when we assume axon to be cylinders, like what we see like here. So that is what this matrix account for. But what we also see is that axons, they are not straight. They can do this kind of undulations. They can be locally crossing, and they can have different shapes. So therefore, all these morphology, 3D morphology influences what we actually believe to be the true diameter. So how could we get insight into these axon diameters? And this is where we use this synchrotron facilities. This is one big one in France, in Grenoble, that I just returned from. And this is the beamline down here that we have. A beamline is where we actually do the measurement And this is where we have all the electrons accelerated with the X-ray, and then we have the measuring station here. So what we did is we took, this is a monkey brain, small punctures here, this is the small holes, and prepared them for coming into the synchrotron. So this is the small samples we have here. Then we put them into this, into the hutch. So we have the X-rays coming in this area here. And this is a big machinery where you put in your samples in a robot, and you have a robot arm. You can see it here. It takes the samples and then put it down here in this high pressure area here where the X-ray will go through and do the imaging. So very shortly how the principle is here. So the X-ray is that we have a big source here. This is the big synchrotron of electrons. So we have a gun setting in the electron, which are accelerated, boost them, and then they come into the storage ring, which is the big one. Every time we have these small things, this is magnets that are bending the X-ray, and every time you bend it, you will have X-ray coming out. This is this one here. And these one here, these are the one that we are using for our imaging. So this one goes into an imaging station, and a long one here make it very coherent, which is important. So this is where the beamline come in into this X-ray. Then we have our sample here rotating because we are doing tomography. And then we have the X-ray going through into a scintillator, which convert the X-ray light into light that is then captured by a CCD camera. So we have two different contrasts here. We have absorption, which is the normal one when we go to the hospital, what we see, where you actually see how they absorb the X-ray when it penetrate. But what we are mostly weighted to in this setup here is because we have a very monochromatic, very aligned, coherent X-ray beam in. Then we can actually measure the phase contrast of how different structures and a nanoscale influences the signal. So we can get a much better tissue contrast to soft tissue. And this is what we are using here. So we're measuring these two contrasts in combination. So this is an example how it looks like. So this is an intact structure. So this is aligned axons and corpus callosum. This is crossing fibers, and this is in the surface of cortex. This is a blood cell, and you can see all the axons, all the cell bodies coming here, and all the axons. The crossing fiber here, you can see laminars of crossing. They're big and they're small. And this one here is a straight in corpus callosum where you can see there's almost no changes. So this is a 3D volume where we just scroll through the imaging, and it's a 75 nanometer resolution without any cutting, any gaps or anything. So when we look into this 3D volume here, so this is in corpus callosum, then we can see many fine details in 3D. So we can see the blood vessels, we can see the axons. It's a bit hard to see here, but you can zoom in. And you can see what we could define as vacuoles, these high intensities here. We don't know what the vacuoles are, but there's a structure which is evenly distributed out the tissue. And we do not see it in all tissue types. So we believe it might be something with tissue preparation it is. Then we have cell clusters. You see it like clusters. This is where we zoomed in. And you can also see they agree what we see on Nissa staining here. So the thing is here that this, you get so excited when you see this in 3D. Until you start to want to analyze it. And that's where you start to have a PhD student, because we are all a bit naive. I don't have time for it, but the PhD student have whole 3 years to segment these anatomical structures. And this is Maya Anderson. And she was so kind to jump into this work, and she's still working with me as a postdoc. So I think she still thinks it's fun. But she got this fantastic result out. So first what we did was we acquired 4 volumes and we stick it together. So we actually get this 612 micrometer long cylinder of structures. And then we can segment all the cells, blood vessels, and vacuoles. And they were relatively easy to segment because of the contrast. So those we segmented with manually and automatic segmentation in combination. And then you can actually quantify all these structures here. And I just forward you to this class paper we have out here. But now we are interested in the axons. So axons have a challenge because they are long structures that go throughout this volume. And these datasets are massive, so you cannot handle the memory. So the first thing we're doing was to extract around each axon here a small image volume. And then we analyze each image volume here for each axon. Oops, each axon here. And this I have a small movie, if it will start. Come on. No. Nevertheless, you will see a very nice movie here, how we actually can segment out the inner layer and the outer layer here of the axons that come through here. And we have like two layers that allow us to all the shapes. So we actually convert this one here up to this a layered distribution of the inner axon diameter, inner diameter, and the outer diameter of the myelin here. So you can actually do statistics. So what we get out of here is that first we segmented out just 56 axons that we could see. So this was a semi-manual segmentation, and it was a very time-consuming process, but it allowed us to look at the shape changes along the axon. And I said that axons are not like cylinders. They have a variation of axon diameter along their trajectory. And that yields all axons. And this is the color coding of the axon diameters that you can see along the axon here. So when you start to analyze each of these axon diameters, then you can see that we get a distribution of diameters. If you take all the axon diameters, variations along the axons, and make one histogram, then you get this red one here. We don't get any small one because our image resolution was too small to see the small axons. But if you take one big axon here, this is the largest one we could see, then you can see it has a larger deviation, variation of axon diameters along its trajectory compared to the smaller one. And you can actually say that the bigger axons has more variation than the smaller ones. So where is this variation coming from? Why? And that is simply because you have structures around the brain. So we have blood vessels, then they take up space. And all the axons you can see in living thing is very, very soft, and that will adapt to the environment of the local environment. So this is an example where axons going around the blood vessels, and you can see shape variations here along the axons. We have other structures in the extracellular space pushing to the axons, also making the shape. So then you can say, okay, that is cool, but this is ex vivo. So it's fixed. What about in vivo? But actually, this is a nice study from Tønnesen et al. So they make a small lesion in the what was it, thalamus here. No, it was counted here in the small region here in a slice. And what they did here? Oh, all movies are not working. What? That's really annoying. Just a moment here. Oh. Can I just... No. Okay. That's
Could
very
it be
pity.
that you have to get rid of the cursor function to
Yeah,
click?
maybe I should try to get rid of this one here. Pen.
Cool to see it.
Here we see
There
it.
we go.
Right. So this is a lesion introduced in this gray matter area. And you can see the timing here. So this is happening live. So you can see the lesion is coming here. And what you will see here is clear cells at some point here. Whoop, they come in moving around to change what is actually going on here. And what you can see, they're moving freely around the extracellular space. So this is a very dynamic space. So they will push around to all the axons, whatever they're actually moving around. And that is part of making these shapes, what you see, what we're detecting it. So yes, we see a snapshot, but it's actually happening in real life. So if we start to look at this axon here, so this is a long axon of around 700 micrometers, and then we can start to see what is happening here. So we can see the node Renoir. That is where the myelin sheets meet each other, two sheets meet, and that is where we have a small, where we have the boosting of the conduction velocity in these small areas where we don't have any myelin. And that's also the regions where we have the smallest diameter. So this is axon diameter, the length, and this is diameter that we have here, variation. What we also see, we have these vacuoles. So this is the axon, and then there's a vacuole here around. And you can see how it pressed to the axon, making shape changes here. What we also see in this one is that when we look at the G-ratio trend, so the resolution is a bit low here, but what you see in the G-ratio is that it follows the variation roughly with the diameter variation. So this G-ratio is maybe something that we should speculate is, what does it mean? It would be logical that the sheet of the myelin is constant within a segment of the myelin, and maybe the axon diameter will vary, and then the myelin sheet will vary with that one. But we see that this is it does not seem to be very independently of the axon diameters. So with this one here is that we could segment 56 axons for a pretty long time. And of course, deep learning can make our life a bit easier. So this is just to show that that we have these large volumes here in 3D. So we need to develop techniques to segment these out in 3D. This is a different way of segmenting with machine learning methods than what you do traditionally. So what we have here, we have this sparse data set here giving us input, and you take small 3D patches here and give it into the network, and you train it, and then we actually do it iterations a number of times so we can increase our training data set, and then you actually get out these nice results here. This is only the axons, but we also estimate the myelin, the blood vessels, vacuoles, and everything here in this 3D space. So this was the synchrotron imaging. From this, we can quantify the axon diameters. So now we have here the diffusion MRI can also measure axon diameters, and this is we showed already for many years ago. So this is an example for in a human. That was in 2007, where we used a simple model here. So we assume axons to be cylinders. It's a one voxel, and you can see even in a clinical scanner, we have axon diameters in the corpus callosum for low, high to low, which is a normal trend of the axon diameter that we expect in these areas here. When we do a scanning on a fixed monkey brain with a stronger gradient, then this contrast becomes stronger. Because with a stronger hardware, we get more contrast to axon diameters. When you compare these axon diameters across corpus callosum, this is the diameters we measure with MRI. We see this trend of small, higher, lower, as what we also see with histology, but it's pretty much overestimating. And we are speculating for many years, how can this be? One thing is that axons, and we also show here, are not straight. It's a very complex form. So one parameter that might influence this axon diameter distribution is that this model here assumes that axons are straight throughout the voxel. But that is not true. And that is why most models today, or models should do, this microcylinder thing. So we make a different assumption, where instead of have cylinders representing throughout the whole volume, we have small piecewise cylinders here, or microtensors here, that are mapped here. They are so small, so they do not see all the configurations of the axons. They only follow piecewise shapes here. That gives a more accurate estimate of the axon diameters that we can use in our predictions. So one thing is here that we do see this variation. So we have these two models here. So this model here with the microcylinders allow you to measure the axon diameters in the whole brain because it's independent on the organization of the microstructural environment. But this, you can only do it in regions where you have straight fibers. This here, this is this cylinder model in the monkey brain where we also saw this high trend in corpus callosum of the monkey. And here we want to explore what does happen if you use different gradient strengths. If it use different protocols, is this estimate actually dependent on this? And it varies a bit, the sizes that we're measuring across corpus callosum, but we can see it more or less coming out with the same trend in the variation of the axon diameters dependent, which is the same trend independent of the protocols that we are achieving. So if we look at simulations, so this is a simulation looking at the different cylinder sizes. So this is sizes of axons. You can see for the smallest one, we don't have any contrast. We cannot see the small diameters. We only start to separate the diameter distributions here when the axons become bigger. And if we have stronger gradients, this is 300 millitesla, then we get even more precise to detect the distribution of axon diameters. So we don't detect the smallest. And this is also something that you can map the sensitivity profile of the axon diameter. So this is the true diameter, and this is the predicted diameters. And this is, we can see here, we have a sensitivity profile. The smallest we cannot see. Here we have some range we can measure, and those we get fail at this region here. So what we see here is that when we start to see diameters, that is because that the diffusion detect cylinders. We need to be in this range here. In this range here, diffusion, the diameters are so small that there's no contrast to diameters. So it's just the model, they predict the diffusion signal as sticks. So it do not have a size. So that is a reason. So we could call this the diameter lower bound. We don't see axons down here. And we also have an upper bound here, which is where we have the maximal axon diameters to measure. Cool. So this was with diffusion MRI. Now we put it all together. So in the same brain now, we want to actually see how these metrics are. So we have the latencies measured with electrophysiology. We have optodynamic where we stimulate. We combine, we need to know the length. We use tractography. We have different measures of axon diameters using electron microscopy. And we measure with diffusion MRI. And we can see the tracers here from injections with fluorescence tracers. And remember, what we need to find out to predict from structure the conductive velocity, the G ratio, and the diameter. So the G ratio we measured, I showed you in previous slides. So this is 0.7. This is what we measured. And to predict the conductive velocity from latencies, we just need to have the length. That is from tractography. So the setup looks like this. So this is what we looked into rats. So we injected a V-racer, a tracer, which is with a CAM2, which is only sensitive or labeling excitatory neurons. It was injected in layer 4, 5, where we have the callosal output of the transcallosal connections here. And this is a staining, fluorescence staining, where you can see the labeled neurons here. And you can see this is corpus callosum. It is layered in the top layer of corpus callosum. This is where we have the projection through these connections here. And we can then take the same animal and we can measure it with MRI and do tractography. And we get out from the injection site to the projection site. And then we can calculate the distances here. So this is the length and the labeling. Then we also have the function. And here we use optogenetic stimulation. So we send in light here in cortex with a fiber. And then we measure here with the electrode, the LFP signal. So this is a local field potential. So when you're measuring, you stimulate here, and then it takes some time for the signal to travel from here to here. And then it slowly builds up because we're measuring the signal in the extracellular space. So it's like an accumulation up here, and then we see a P1. That is where we see the peak, where we have all the maximal signal arriving. That is our latency from stimulation to here. Then we will have in the onset of N2 peak. This is the pink, and then we have the green, which is in one the negative pulse here, which is something else when we have reactions in the cortex. So we have 13 animals here, and we can just see how the time delays. What is interesting is here that for one animal, the line here, if it's short time here, it will also be short up here. So that it's just a quality check that what we're measuring makes sense. What we now do is we put all this together. So this is the big drawing here. So we have our optodynamic, this is our latencies, what we have measured here. And we have the electron microscopy on the same animal here. And that is using the standard EM with the diameters we have predicted. And we have the cryo-EM. This is just one animal, but we have more coming up to compare. And then on the same brains that have been with electron microscopy here, we have the diffusion MRI axon diameter estimates. Now we can put this in one structure function relationship protocol because we know there is a linear relationship between conduction velocity and axon diameters. And this is the two axes. So we have the conduction time or the latencies here, which is 15 milliseconds down to half. And that agrees with the axon diameter. The bigger axons, the lower conduction velocity, and the smaller axons, the slower conduction velocities. So what we can do now is when we compare what we see at the electrophysiology, all animals here are placed in this period here. And this is a zoomed in region. So they are all between 7 and 4 milliseconds here. And we see if we make this line down here, these latencies here fit to the size of the mode of the axon diameter distribution, meaning the smaller axons. When we start to compare here, we can also see that we have the cryo. We have the peak coming up much later here because we know the cryo is not really affected by shrinkage. So that means that it actually predicts that the conduction velocity should have been a bit faster. But this could be that this is accumulated latencies we're measuring here. So there might be some variations. But nevertheless, if we apply shrinkage correction to these axon diameter distributions, then we push the diameters down to these striped lines. So that agrees very much what we see with cryo-EM if we compensate for this shrinkage. Nevertheless, it's meaning that the latencies here measured with LFP is to the small axon diameters. So if we now look here into, if we predict now the latencies with the diffusion MI. So we see here, this is for the number of animals we have. So it's a very broad peaks because there's some uncertainty here. They are very large. The diameters, if you compare it with the peaks here, what you see here with the EPON, with the EM, and that is because with diffusion MI, we are not measuring the diameters, we are measuring with EM. It's a volume-weighted diameter because it's a 3D measurement. So that pushes the mean diameter up to larger values compared to what you only see it when you make the cross sections here. Nevertheless, you can compensate for this. So you can add this volume weighting to your EM result, and this is this one here. So that pushes the diameters, these mean volume weighted diameters up to these ranges here. And if you map them down here, then you can see it somehow fit a bit to what we see with MRI, but the MRI are still larger than what we expect here. When we compare with the cryo-EM, we can see we push it to higher diameters here. And the reason why these two agrees is because we only included diameters in this from this cryo-EM that we know is detectable by MRI. MRI do not detect the smallest diameters, so they are not included it. But what we can see here is also when we compare is that these large axons here, no matter what we do of shrinkage correction and whatever, the conduction velocities, the latencies measured here with LFP does not match with the diameters that we are measuring with MRI. So that means that MRI is measuring a different regime of conduction velocities that, for example, the LFP signals here are detecting. So putting it all together, so we can predict this structure-function relationship. So different modalities have different sensitivity ranges to the predicted relationship. And this is what we show in this figure here. So we need to be careful when we are actually start to comparing the prediction of structures to functions and the others, and the same thing from function to structures. So what we can see is that axon diameter distribution is the one that actually are covering the whole range of what we could expect to be the conduction velocities. But and that's axon diameter. But what we need to be aware of here is that we need to ensure that the tissue quality is high, and that the tissue processing sometimes may introduce shrinkage factor. So that need to be compared need also to be adjusted for when we are comparing with the latencies that we are measuring. What we also need to be aware of is that the microscopy is a direct analysis. We see structures directly, whereas MRI is a model-based estimate. So we need also to be aware of the different ranges of axon diameters that these two metrics actually are measuring. So we also need to be aware that axon morphology is a 3D structure. And what we also have shown is that even that is 3D structure, then if you take matrix of 2D histograms of axon diameters and compare it with the 3D, you take all the measurement changes along the axon and make it for population of axons, these two will be similar in distribution. So that means that we actually can use all T2 axon diameter histograms, what have been done for many years ago. But we need to be aware that these metrics here, they are affected by the morphometry of the axons. So that is what you can handle with 3D structures. And they're not straight, and they are crossing. What we also need to be aware of is that these local field potentials, which are predictions of this P1 that we expect to of the latency, what we showed here that is related to the smallest axon diameters. So if we want to detect the sensitivity to the fastest one, then we need to design our latency matrix that can detect these faster conduction velocities, or the detection of the fast one. That simply means that we need to have a method that we can go down here as soon as we see starting here, so that it can be individual cells that we can measuring here to be sensitive to these features here. That was it. And then I just have an acknowledgement here. So that is for all the people that have been contributing to this. That is many people over the years. And what I've been presenting here with all this, with the electrophysiology and all this is my previous PhD student, Christian Skoven, who did a wonderful work here. And then, of course, we also have collaborators, which is Harvey, but also specific Maurice, who provided the monkey stuff that we have, and people at the synchrotron facilities that has been spending hours and hours and hours with us collecting these data. And then all the funding, and specifically the study here, was supported by my ESC consolidator funding. So, thank you.
Amazing. Thank you, Tim. This was every time I see your talk, I'm absolutely mind blown what you put together and the level of detail that you can see. I want to open for questions to the floor. Raise your hand so I can see who has questions. Also for people over on YouTube, if you have questions, please write them and I will bring them over here. I'll get us started for now with, I have so many questions for you, but let's start with the fact that you said that the axons aren't straight and they're crossing. So especially when we look at the corpus callosum and the midsection, there is this perception that they all nicely align, and you've shown in some of the slides that they seem to layer up. So you have axons that only go through the top, which then I assume means that others go through the bottom part. But also that they're not as straightly aligned as one might think. So I was wondering if you could tell us a little bit more about that.
But that's true. So first of all, how the organization of these axon fascicles depends very much of the species. So what I showed with the fluorescence imaging, that was in rat. And in rat, when we have the corpus callosum, the different projections of cortex from one side to the other side, they are organized like on stacks, like a sandwich on top of each other. If you go to the primates, then it's a different organization. Then you actually turn everything up. So when you have from right to left, they will do like sheets like this. And so that is just a different thing, but organization between axons. We have a eLife paper out on this topic here. But when you zoom in and you look at it in 3D, you see small axon bundles, fascicles that actually completely have different ways in the corpus callosum, even that you expect them all to be straight. They typically follow
completely to the point where they orthogonal to each other even?
Yeah, so they are very, yes, that's a different story I did not talk about here. So there seems to be an organizational rule of how fibers they are crossing. So first of all, they are organized similar. It looks like a kind of lamina, it's like sheet. What have been shown in a science paper with Van Dien for many years ago that have been a big debate for tractography. But it actually seems to be that this is actually not. It might be some truth in this one. We see the same thing in this synchrotron in different bundles here. And it is that if you have two bundles that are following the same way, they will have like follow each other very in parallel. If you have fibers that are crossing, then they actually, for reasons when axons are formed, is that they actually will always try to follow each other almost on a grid-like sheet. So that is what you see. Yeah, so I don't know what more to say. So when they cross very much on a grid-like structure with a high angle.
Amazing. So that's very interesting. So the science debate that you refer to with the sheath versus the argument that is more like the Victorian streets of London, is that a principle of organization that you see specifically when it comes to callosal fibers, or have you also observed similar patterns elsewhere?
No, it seems to be much more general.
Okay.
So we also see it in crossing fiber regions and different parts. So these are small samples that we take. This is these synchrotron experiments where you can take these small samples here, and then we can actually map the structures in 3D. And if you take different samples in the brain, it's not only in corpus callosum you see this laminar organization. It seems to be a general trend, and it has different ranges depending on where it is crossing. But it seemed to be organized in this way.
I'm going to hand over to Anna.
Hello, thank you very much for this interesting talk. Just a very general question for all the imaging people here. What would be your key takeaway for us relying purely on diffusion data or fMRI data, MRI data? Shouldn't we trust it as much as we are doing, or what would be your advice to keep in mind?
I cannot tell about if that's not my, so I cannot give advice on that. But I can tell that yes, you can trust it. But things are a bit more complicated. So I think the problem is with diffusion MI, it's sensitive to the microstructural regime that we actually have limited knowledge about, especially how it looks like in 3D and how this 3D organization actually influences the geometries that we are looking at. And with this synchrotron actually allow you to have a certain size of a field of view, so you actually start to see on almost on the voxel scale what is happening in this organization here, and put all these small puzzles together and actually start to give a picture that makes sense what we are measuring with, for example, diffusion MI. So I think you can trust it, but also need to understand that it's not the same.
Right, right, and probably would visualize way more if you would take that into account as well on top of yeah, imaging purely.
Yeah. I think what it is what I'm saying is that it is the interpretation of the results, where we need to know more about how does this anatomical on the micro regime actually look like to understand what is it actually that we get out with MRI.
Right. All right, thank you.
Eduardo has a question.
So thank you very much for the presentation. So mind-blowing. I have a question. So it relates to some distant memories about neurophysiology course of the central nervous system. So as you mentioned, one of the main factor that can be used to predict the velocity of conduction is the axon diameters. But if I remember correctly, there is also the numbers of Ranvier nodes that is an important factor as speeding up, like the more the nodes, the higher the speed. And I was wondering whether this can be captured or how if that fits into your research.
Yeah, so there are many parameters that that are influencing the conduction velocity along the axon, and I showed it on the first slide. And we cannot handle all this, but there seems to be a relation with the length of the node and the thickness of the myelin is how long and determine how well isolated the signal is when it is jumping between nodes and Ranvier. And this relationship describes the length of the node and can also have influence on the conduction velocity, but that is not something that I've been looking into, so I cannot tell. But I know there are studies that have been showing different relation between conduction velocity and the size of the node.
Oh, okay. Yeah, thank you so much. Yeah, that came to my mind when you mentioned that the approximate linear relationship between the diameter and the velocity. So yeah,
That's
okay.
true. So we looked at what we believe to be the main factors, but this is the beginning. So I hope people
Yeah.
they will top up the new information.
But would it be correct to assume, for example, that in the measures that you have of the diameter of the axons, also the sheet of the myelin is included, right?
Yeah. So what we're measuring here is we are measuring the size of the axons in the same area that we're measuring with the conduction velocity. So in this, I cannot was transporting the signal. We only have the distribution.
Okay. Yeah.
So that also influences this matrix here. But still, you have the full distribution and we have something and we know a relationship, so we can actually start guessing up what should be the size relation between the sizes versus the latencies that we are measuring and predicting.
Thank you so much.
Mm-hmm.
I have another question about the postmortem imaging. So you mentioned that there is a shrinkage that on average is about 40%, which I find absolutely amazing that we have such a change in the postmortem imaging. And in the field of tractography, where we do postmortem imaging of the brain as well, the Rachel Barrett, for example, has shown beautifully that rehydration improves the signal. And I was wondering your shrinkage correction, if how you do it, what you use, and if you also rehydrate the tissue samples.
So there's two different things. For when we do postmortem MRI, then it has been shown for a long time that formaldehyde binds the water. So therefore you need to rehydrate. And so that is to reestablish the signal, so we get more signal when we are doing the postmortem. And we always do that. And I think most people, they are doing that. That is different from the shrinkage. There might be some shrinkage due to the fixation. I cannot tell you how much, but there is some. I think it also depends on how long time the tissue has been fixed and different things. But the shrinkage, the severe shrinkage, is coming from when you're comparing with tissue samples that you're prepared with dehydration, because you're drying out the tissue as a process before you do the staining, or as part of the staining procedure. And that is where you introduce this kind of shrinkage in the traditional way.
I see.
And then, so you compare something that you think is validation, but you actually introduce a stage where you actually manipulate with the space that influence. So you are actually measuring two different matrices: postmortem with MRI, in electron microscopy, which have this shrinkage factor, and that you need to adapt.
I see. Thank you.
Many people have been reported this shrinkage over the years and guessing. So, but now we have also the cryo to make it. We try to make it more precise. But it's just to start the discussion here.
Now, you started a discussion because I actually invited someone who had many questions for you over on YouTube to join us live in the chat and ask all her questions. So, Nicola, take it away.
Hi, thank you, Steph. Thank you for sending me the link. Tim, a beautiful talk. I followed it via the YouTube live channel. So I work in Jülich at the research center, and I do post-mortem analysis, basically receptor architecture, but I've also looked at cyto and myeloarchitecture, and also we work with polarized light imaging images, right, to look at myelinated fibers. And in these PLI images, I've seen things like those undulating shapes that you mentioned. The problem is that it's not something that I see consistently throughout sections or that I can follow. So that's why I tended to write them off as artifacts. I'm just wondering, because you mentioned that axons with a larger diameter tend to shrink more strongly than axons with a smaller diameter. I wonder if this undulating pattern could maybe be associated with the fact that precisely at that position there's maybe a change of multiple axons that are suddenly, diminishing their diameter. Because so I've always mostly seen it in more superficial parts of the white matter.
That is not what we see. So the undulating, I think, I presented this undulating for many, many years ago. I showed one of the historical slides where you actually can see this undulating. It's not a general trend that you see in the brain. You see the undulating very clearly in the PNS system, because it's a mechanical effect where you actually can stretch the neurons. You don't have the same need in the brain. So it's by coincidence that this is, I think there might be something, but you don't see undulating. But they do something, but it's not undulating that like you see a sinusoidal structure.
Right.
So the bigger axons, when we look at in these 3D images, is that you can see this this shape arranges, and it does not look like you see like a local area where it shrinks more than others. And there's a reason for that. First of all, big axons, when you look at electron microscopy, they are always spread out. They have a low density, and the density is equally distributed within the field of view. You
Right.
don't see a population of big axons, just one bundle with big axons. That is very rare that you see that. It's already intermingled with different sizes. And you do see that these have more shape variation. So we hypothesis, or we speculate, is that when you have big axons, and we have a bioarchive paper out now on this topic that I've been talking about, and it hopefully will be out soon. But the idea is that when you have big axons, inside the axons you have a cytoskeleton. And there's more free space in the big axons than the small axons. And this might be the reason why when you do the dehydration is that you will then see a change, larger change in the larger axons than in the smaller ones, simply because of this maybe mechanical stabilization inside the axons. This is what we sort of find in the literature might explain this difference here. But they are much more severe to shape changes than the smaller one.
Thank you. And then I have another question, the one concerning the crossing axons. Do you find them everywhere inside the corpus callosum, or are they closer, like to the interface with the cortex, or closer to, well, you know, if you're, for example, to this to where you'd expect to find a cingulate bundle?
So it needs to be remembered that what I've shown is small samples in corpus callosum. So we generalize from these small samples. What we see is that you can have these small local fiber bundles, or they can be consistent of few axons. It's a small one that can simply just have an orthogonal trajectory than the rest of the pathways. For example, in the mid-sagittal corpus callosum, what we see. Often these tracks here, then follow the blood vessels.
Right.
So because that's like space around to have it. Why they're there or whatever, I cannot tell you. But you have these crossing fibers, and they are crossing like very. It's not. They have a high inclination angle in that way.
Thank you.
Was that all your questions, Nicola? Felt like you had more.
Well, no, look, there are other hands up here now.
Right, okay. Ahmed, take it away.
Thanks. This was really enlightening. I have a question about how this could fit into our understanding of diffusion signal better. So you've done so much incredible work on modeling things at such a small scale, and now and in 3D, which is really great. And I think this could offer a great opportunity for us to understand better how this could translate into, through simulations, into what a diffusion signal might look like, knowing the underlying structure and how that then links to what we measure with MRI. Do you have any idea how that could be done, and if you have any plans on doing that?
Yeah, so we actually have done it. So what we learn from this density, what we learn from these 3D volumes, is that we can get a statistical matrix about how much are they, we call it microdispersion, how much are they spreading out, how are their trajectories, how are big axons varying in size in different regions, and why are the size changing because of cells. These things here are things you can put together, and then you can make a microstructure model of a synthetic environment with cells and axons, and then you can actually generate the statistical representation synthetically that we see in this synchrotron imaging. And we have just published out we call it a white matter generator that can generate these synthetic axons. And what is
Right.
shown
Okay.
in that other groups also doing this kind of things where they also actually account for the shapes, where the shapes are coming from the interaction from the different neighboring axons. People have also been doing simulation in axons from electron microscopy, 3D electron microscopy, and we have also been doing in this synchrotron imaging, where we benefit, the synchrotron benefit for the large field of view, whereas the electron microscopy often has a very small field of view, but then it can see some of the smallest diameters that we cannot see. We are predicting these. So the different techniques give different opportunities. When you do this kind of simulations, then you just need to be aware that you have a size of the geometry that is big enough to sample the structure without being conflicting that the structure's ending. So it needs to have a certain size to really have the diffusion process to really detect these different shape changes that you see. For this synthetic generator, we have included that you actually can move around cells. So you can actually do the same thing with what I was showing. So you can generate your own shape. And it should actually map with the statistics we see, morphological statistics. So you are absolutely right. This can be used for us to learn more about how we actually should do this. My personal interest is the dynamic of white matter. That is like the next step. I think we have good models for the axon diameters and understand it pretty well. Now the next step is to include other kind of features.
Right. Okay. That's great. I'm gonna look that up. Thanks.
Yeah, you can do it online. It's also on a yeah. You just try it.
There's another question from Li Ting.
Yeah, hi. I have a questions on so you mentioned that these scanning all like this data are building from rats and from monkeys and from post-mortem brain, human brain. And so I'm curious how far are we to also construct apply this method onto more large scale human living human beings.
To living human or just to whole human brains?
Or whole human brain.
Yeah, okay, because it's a big difference. There's a lot of interest to be able to the synchrotron imaging technology is a very fast moving field. And as I showed it, you can actually make these small samples now, and you can scan this nano resolution. And there's also been introduced what we call the HIP-CT, another kind of setup where you actually have a whole brain where you can zoom into the small features, and the resolution there is down to around 2.5 micrometer. So it will be possible in actually a pretty near future that you can map out the whole brain in this very high resolution. But that's not the problem. The problem is to handle these data sets, because what I showed is very high resolution data sets with massive data. So the whole infrastructure to process and handle and analyze this data is very different from what we have done so far with MRI or any kind of big multi-center scale data sets. So I think it will be possible, but it will be challenging to process the data.
You will need a lot more PhD students.
Yes.
Nicola has another question for you.
Yes, this doesn't have to do with axons, but it has to do with the data that you just described, entire brain resolution down to 1-2 micrometers, or even if it's only 5 or like only 5 micrometers. White matter neurons have their existence has been controversially discussed. Do you see have you come upon white matter neurons when you're mapping the axons? Or something that could be that could be interpreted as a white matter neuron? Because then, if so, I might want to ask you if we could share a PhD student that might want to not look at axons but look at white matter neurons.
Yeah, I know that's been reported, this white matter neurons. I have seen many things in white matter. And I cannot say specifically if I've seen neurons in white matter with this one. So I cannot tell you. I've seen different things.
Okay.
Yeah. So
right. So, but it's not just like a sea of axons. There are, and not just axons and blood vessels. There are other things there.
Yeah. True.
That's
But it
the
depends on
need.
what area you are in the brain.
Yeah.
Yeah.
Oh,
But it
exactly.
could be interesting to look at. I agree.
Thank you again.
Love the public trading of PhD students here.
But they are really important. Otherwise, you cannot do this things here.
That's very true. It's very true. I don't see any more hands raised, so I think we're ready to let you off the hook and give you a bit of a rest. Thanks for presenting your work. Absolutely fantastic. Looking forward to hearing more and seeing more in the coming years for the preprints that you mentioned. Thanks for taking the time, and it was absolutely well worth waiting that week while you were playing around in Grenoble.
Yeah.
Thanks for joining us. Thanks everyone for being here. The next talk is going to be end of February, the 26th, I believe it is, with Sahba Bejarati. See you then. Bye.